Hello all, welcome back to Nanotechnology in Regenerative Medicine. This is Unit 5 where we will be discussing about the nanotoxicology. So the course outcome of this unit would be to understand the issues related to the toxicity and environmental impact of the nanomaterials. So to get an overview of what nanotoxicity is, in general terms, any toxic effect caused by the nanomaterials that are basically classified when the size falls somewhere between 1 to 100 nanometer in size. So when they interact with the biological systems, including the human cells, tissues, and organs, and the environment. In the environment, it would include all the system like air, water, soil, and animals included. So animals, any toxicity caused to the animals would come under environment. But the toxicity caused to the lab animals, that is a rat model or mouse model, that would come under the normal physiological toxicity. So if we have to define the focused environmental nanotoxicity context, it is any harmful effect caused by the nanomaterials on the ecosystems that includes soil, water, air, plants, microorganisms, and animals, as I said. So when they are released into the environment, that is how would they be released? That is through the industrial effluents, or sometimes the photocopy machine would also send out some soot. So these are all the different ways. These are two of the different ways how the nanomaterials escape into the environment and affect any of these factors of the ecosystem. Now let's dive deep into each of these concepts and try to understand the mechanism of how toxicity is integrated. Let's assume this being a human cell, which would be in the size of around 1 to 10 micrometers. However, when you look at the size of a nanoparticle, this is 1 to 100 nanometer, which is more than 1/100th of its cell size. So they can easily penetrate into the cells. That is, they are invasive. So when you look at the size of these nanoparticles, they can easily penetrate into the cells and interact with any of the cell organelles and can cause damage, metabolic damage to each of the cell organelles. So they can rupture the cell membrane. That itself is a problem, which we have seen in LDH assays in your animal biotechnology lab as well. If that did not bother you much, the nanoparticles can also interact with DNA and genetic materials, causing epigenetic modulation or even genetic modification as well, which is so permanent. So there will be some change in characteristics, which you may expect that you might become a X-Man character or a Spider-Man, but no. You may end up being Stephen Hawking's, except with less cognitive ability. So nanotoxicity is worse than what we know or what we assume. However, even though it is not completely well established by the scientific proofs, but this is a very high possibility. And this can definitely happen because any nanoparticle is a xenobiotic, and we don't want a xenobiotic interacting with the DNA. Now, let us discuss what gives toxicity to these nanoparticles. The major issue is the surface reactivity, where the free electrons from the nanoparticle surfaces can react with the first thing it sees on its way, and also either the positive or negative charge that is highly electrifying. So that can also harmfully interact with the biomolecules, leading to metabolic degradation. The next major issue is the bioaccumulation of the nanoparticles. The nanoparticles sometimes overstay in the human body, so that can attract the unwanted immune cells towards the organ where they are accumulated at. For example, if the nanoparticles are accumulating in the liver, they tend to attract a lot of immune cells, antibodies towards the liver. So that can cause the immune cells to attack the liver cells as well. So that becomes a problem. So that can lead to autoimmunity and hypersensitivity issues. So bioaccumulation is a serious trouble. However, as I said, the scientific proofs are still a little limited. The reason we will discuss in the class. The next toxicity mechanism is how our body reacts and responds to the entering nanoparticles. There is this issue called protein corona formation, where the proteins identify the nanoparticles entering into our body. Some proteins, especially the lipoproteins, they identify the xenobiotic and they tend to form a coating over the surface of the nanoparticles. Now, the problem associated with this approach is that they tend to go and attach to the surface in a hurry so that they even skip the, the checkpoints where the 3D confirmation is perfect or not. So that leads to protein misfolding, which leads to unwanted protein accumulation in the body. And since now the surface has become biocompatible due to the protein corona formation, these nanoparticles easily enter the cell, which causes degradation to the cell organelles, and these can also trigger the immune response. So, protein corona formation, which is a physiological response by the human body, that can still cause toxicity. Now, to understand why Arjun Reddy is toxic, sorry, why nanoparticle is toxic, we need to understand the intrinsic properties and physicochemical properties of the nanoparticles that gives this toxic attribute to the system. The first thing is, as we know, the size. The smaller the size, the more penetrating it can be, the more invasive it can be. And the surface charge and surface reactivity as well, we have seen about this for a very long time. And the chemical composition. The chemical composition of the nanoparticle can be either inorganic or organic. Benzopyrene, I was talking about in the last class, that also is an organic nanoparticle. And the aggregation state. Some nanoparticles, especially the colloidal nanoparticles, tend to aggregate in large amount inside the body. So that can severely damage the tissues and organelles. And bioaccumulation, which we saw just few seconds ago, can cause a lot of issues, including autoimmune diseases as well. Now, one other important issue and interesting issue is the exposure, the route through which the nanoparticles are entering into our body can determine whether the nanoparticle is going to be toxic or not. Some nanoparticles that are not invasive dermally, that is, when we use it in the skincare or other ointments, even though they are not toxic dermally, when they are inhaled, that can become a problem to the pulmonary tract. That can accumulate in the lungs and cause unwanted effect on the body. Some nanoparticles that are palatable, which we conceal in a capsule, should not be go in contact with eyes, or it should not be inhaled. So that can travel right into the brain and can change the chemistry forever. So the exposure route is also very important. Now, there is one short video regarding the scientific experiment of how nanoparticles entering the lungs can accumulate there and go to different parts of the body and cause toxicity to these organs.
Tiny nanoparticles can, in fact, nestle themselves in our bodies with a single breath. The question is, how long do they stay there? A UK study suggests that the long-term accumulation of nanoparticles in the vital organs of rats warrants a closer look at the bodily effects of nanomaterials. Most animal studies have shown that breathable nanoparticles that are lodged in the lungs are typically cleared within a few months, with fewer than 1% traveling to other organs, such as the brain, liver, or kidneys. But what happens to these particles long term? To answer this, the UK researchers tracked iridium particles inhaled by rats for over 3 months. During the first week, their results seemed to agree with those reported in other studies. A small fraction of particles accumulated in the kidneys and liver. But in the liver, they appeared to pile up indefinitely. And what's more, the particles were cleared much more slowly from the lungs than previously observed. Together, these findings suggest that continuous exposure to nanoparticles could present a long-term toxicity risk. But before regulators slap warning labels on products containing nanoparticles, researchers will have to address a few issues. First, they'll have to prove that the nanoparticle pathway observed in rats is the same in humans. They'll also need to extend their study to include much more common nanomaterials, such as titanium dioxide and carbon black, found in cosmetics and paints. While no imminent danger has been proven, least of all the threat of a ravenous gray goo, the researchers' findings are telling. At the very least, they provide grounds enough for doubling efforts to examine the effects of nanomaterials on the body.